My name is Grace Artis, and in this video we'll be comparing the RNAi and CRISPR mechanisms. Let's say we have a eukaryotic cell that produces a protein from gene A, and we want to investigate what happens if we eliminate the expression of this gene. Which approach should we use in order to do this? RNA interference is a precise mechanism used by eukaryotic cells to censor the expression of genes. It is triggered by double-stranded RNAs which bind to an enzyme called dicer. Dicer cuts the RNA into tiny segments that are unwound to produce small interfering RNAs and/or microRNAs. These small RNA molecules are only about 21 nucleotides long. Both siRNA and miRNAs combine with proteins to form an RNA-induced silencing complex, or RISC. The RISC pairs with a messenger RNA molecule that possesses a sequence complementary to its siRNA or miRNA component, and the RISC then either cleaves the mRNA, leading to its degradation in the case of small interfering RNA, or represses translation of the mRNA in the case of microRNA. RISCs with siRNA are very precise, often being the perfect complement to the mRNA sequence. Conversely, miRNA only partially complements the targeted messenger RNA, but this allows for the miRNA to target many different messenger RNAs. CRISPR stands for clustered, regularly interspaced palindromic repeats. This mechanism is used by prokaryotes to protect themselves from invading DNA. CRISPR is not only limited to prokaryotes, it has been adapted to allow for genome editing in eukaryotes and has even been used to target HIV. In this mechanism, guide RNA is produced to match the sequence of the DNA to be altered. The gRNA then forms a complex with a protein called Cas9. Once the guide RNA finds the matching DNA target, it is then cleaved by Cas9. The DNA target will attempt to repair the break itself, but this often leads to many errors and the gene could be disabled. Another option is for a piece of DNA with the desired sequence to be inserted, which allows for precise gene editing. So which method should we use to eliminate gene A in the eukaryotic cell? Though both are viable options, I believe it best to go with CRISPR in this scenario. Since CRISPR is extremely precise, it could more easily target gene A. Also, once the gene is knocked out, this method could be used for further investigation into gene editing for the cell, and gene A could be altered in any way desired. I did not choose RNAi because due to the nature of its imperfect base pairing, miRNAs have a high degree of variability in regards to what mRNAs can be targeted. Though siRNA still perfectly pairs, I believe CRISPR to be a more precise method for gene elimination, as well as having more options for further research into gene A's functions.